immune system.
Excuse me, madam.
Yes.
Kotwal, madam.
Yes, ma'am.
Excuse me. Yeah,
Yes,
voice
ma'am.
is constantly cracking.
Okay.
Your
Shall
voice
I
is
change
constantly
to
cracking.
okay. Now also you are not audible.
Yeah, it's constantly breaking.
Hello.
Every word is breaking. Every word is breaking.
Okay. I will escape and I will change my network. I'll
Okay.
change
Okay.
my
Students,
network.
please wait.
Huh.
Students, please wait. There is a disturbance in voice. So madam is changing her network. Till then, be patient.
Okay. Can you see the screen? I have changed my network. Now I have come on Wi-Fi mode. Because if the electricity supply is disturbed, so that's why I was using my data. But if there is a problem with the no, voice, then I have changed my network also. Can you hear me? Am I audible right now?
Yes, ma'am.
Yeah, ma'am.
Yes.
Uh,
Yes, ma'am.
And see, the only the problem is there if there is a power supply cut, then I will be out of the station. Just madam will inform in that case. Okay. So these are the different branches where we can use this biotechnology. Now these are some of the examples. Children, now these are very different examples which you might not be knowing also. Some of the examples are very new to you. So note it down way as and when I tell you something. Okay. So these are some examples of therapeutic products made by recombinant DNA technology or techniques. First is blood proteins. First example is erythropoietin. You know what is the meaning of erythropoietin? Now try to understand the meaning, then you will remember also. Erythro is the word related to blood cells. Okay, poietin is generally the word which means formation. So red blood formation, that that means formation of RBCs, which is related to this, is one of the important factor which can be considered as a very important example, that is the erythropoietin, where if there is any problem with the kidney system, generally in that case what happens, this erythropoietin gets affected, and then because of that there is less formation of RBCs, and which results into anemic condition. Okay, note it down somewhere in the this thing in your notebook. Okay, so this is one examples where then erythropoietin formation can be done using this rDNA technology. And factors, what are these factors now? These factors which are mentioned here as the same problem is there with this again. I'll just try. Tissue plasminogen factor or urokinase. This is now again you can try to understand. Okay, factors like factor 7, 8, 9. These are related to again blood related factors. Means this is actually hemophilia related factor. What is the hemophilia? What is hemophilia? No, the hemophilia is the genetic disorder that occurs because of the defect in the gene, and which is inherited from one generation to the another generation, and these are the factors of the gene called 7, 8, 9, and because of that accordingly the name is given. The name is given factor 8 hemophilia A, hemophilia B, where there is a problem in the clotting of the blood. Okay, so because of the defect in the gene, the clotting of the blood doesn't take place, but it gets affected, and it affects the entire system, human system. So this may cause, if there is no formation of, if there is a blood clotting formation doesn't take place, then what will happen? It will result into the loss of blood. So in case there is injury to the body, then continuous flow of blood will be there, and so it will not get clot, and then the body will lose a lot of amount of blood that may result into the death of the person also. So these are the factors. Then tissue plasminogen factor, you try to understand tissue related to the tissue, the cells, a group of cells, and which can be related. You can relate the meaning with the this word. Okay, urokinase is one of the factors. Here, this is nothing but a serine protease which is involved in many biological functions like fibrinolysis. Fibrin is what? Formation of a fiber. Lysis means what? Destruction or killing. Then the five when it this is again related to the blood. Then if there is no formation of the fiber, and whatever the fiber is produced, then if there is the lysis, that means the breaking of the fiber, then this in the other case can be helpful. How it is helpful? Then if the clots are produced in the body, in the blood, then it may affect the flow of the blood. So in that case, this plasminogen activator is can be produced using this technology. And urokinase is this type of enzyme, that is the protease, serine protease, and which is involved not only in the fibrinolysis but embryogenesis, then cell migration, tissue remodeling, wound healing. Especially when I was telling about the tissue, wound healing can be done with the help of this activator. Then next is human hormone, that is the epidermal growth factor. You know, epidermis means outermost layer of the body, which is considered to be the any organism. Okay, which that growth factor, if for wound healing and such injuries, or if any surgery is done, in that case also it is very important thing, so easily that healing can be done. Then follicle stimulating hormone, FSH, you might have heard, which is related to the reproductive system of the human beings, which is involved in the formation and the no, not formation, development and the functions of ovaries and testes. It is also involved in the control of menstrual cycle, so it helps to control the menstrual cycle. So this is the FSH factor. So these are very important hormones. Then again, insulin, you know, everybody, the example insulin, where what is the role of insulin? Which maintains the controls the level of sugar in the blood. That is very important. So such type of hormones can be produced using this technology. Then immune modulators, these are the different examples of immune modulators like interferons, alpha, beta, etc. This interferon, you try to understand the meaning of interferon. These immune modulators that are related to the immune system. Now you know what is the immune system of the body. What are the cells generally involved in the immune system? WECs and few more cells of the body, they are involved in the immune system, right? If there is infection or the attack of the virus to the host, then what happens? That host cell prepares or produces this modulator, or this is a type of protein, you can say, which gives signal to the host cell and interferes the activity of that virus which is attacked. So what is the role of virus here? The virus which attacks to the body, it stops the functioning of host cell, and it starts its own synthesis of genetic material, right? This is what generally happens. Then in that case, these interferons, these proteins which are produced by the host cell, they interfere the activity of that attacked virus, and in that way they help to control the and increase the immunity of the body. Just like interferon, then colony stimulating hormones, they are also related to somewhat a kind of blood cells. They are also helpful in the immune system of the body. Then tumor necrosis factor. Now one more thing you just remember here. Tumor, what is the word tumor indicate, and where you might, almost all of you might have known about this word. Okay, what is this word? uh, this word tumor means excess of growth of the cells, no, which results in the formation of knot like structure, or you can say it as a tumor. Okay, that is related to what? That gives the excess of growth of the cells, that is called as cancer. You can say that that tumor can be cancerous or non-cancerous also. That tumor is nothing but the excess of formation of cell. Okay, necrosis means, in general, the word of necrosis is what? destruction or death. So death of these cells takes place. So the factor which is responsible for these cells which cause the formation of tumor, that can be produced, that factor can be produced, that means tumor necrosis factor can be produced using this rDNA technology. Are you getting my point? Tumor is what? Excess of growth which results into a knot-like structure that gives the appearance of a tumor, and necrosis means what? Death or the division or the destruction. So destruction of this tumor can happen using the factor. So this is related to the cancerous cells. Okay, so this is very important, related to the cancer type, different types of cancers. Then along with that, different types of vaccines are produced. So what are these? Some of the examples are just mentioned here. Vaccines like cytomegalovirus. So cytomegalovirus, for this virus the common host is human beings and monkeys, and these very commonly it is observed in the human beings and monkeys. So what happens generally in this case? Cytomegalovirus, what it how it works, when it affects, injects or affects the body, the uh, entire gastrointestinal system that gets affected, and it starts from the face, then the intestinal tract, and then it causes different types of ulcers, then liver inflammation, then intestinal obstruction, colitis, etc., etc. So in this way it affects the body. So vaccines are produced against this and using this recombinant technology. Then hepatitis B, hepatitis A, everyone knows hepatitis B, right? Yeah, you must the common word, or in Marathi what we call it as kavi, right? So what is this hepatitis B? In Marathi we call in general, the word which we use for this is kavi. What happens in this? Generally symptoms of this are yellowing of the eyes, abdominal pain, and dark urine, right? So this is all observed in this. Okay, so then in that case what happens? um, Generally, this yellowing of eyes and abdominal pain, if the infection is chronic, in that case, what happens? Liver failure is also observed, and also sometimes the cancer and scarring also can happen, is observed. So this is what one of the applications, or where vaccines can be produced against such diseases. Then measles, what we in general call as gower, right? So this is the highly contagious viral infection, and generally muscle pain, fever, then dry cough. And the most important and the uh, common observed, commonly observed thing in this case is what? That this is um, rashes, skin rashes. What is the skin rashes? Generally, what it results into is the rashes also in the next stage, like later stage, what it gives into that, or flat red spots in the face, that results first on face, then neck, then remaining downward body parts it is observed, and then these small raised bumps also may appear in some cases in the later stage of the measles. So there also vaccines are produced using this recombinant uh, biotechnology. Okay. And the next example of this is rabies. Rabies vaccine is produced under rabies. What is that rabies, and when it is observed? That this is generally uh, observed by the in saliva of the infected animal, and the common animal is what? The common animal is what? Dog. Okay. So in that case, what happens generally? This is the deadly virus sprayed to people from saliva of infected animal like dogs. Okay, and what are the symptoms generally? First symptoms are fever, then headache, then excess of salivation is observed. Then if it is in the next stage, what happens then? Hydrophobia. You know the meaning of hydrophobia? That is fear of hydro, that means water. Now, few more examples which are given in the textbook in a very good manner they are mentioned, and that you can remember also. That means in one column disorder, disease, or the health condition is mentioned, and the other the recombinant DNA technology, how it is used to overcome this disease or disorder it is mentioned. So you learn this, it's very important, and it is going to help you, which relates to our daily life. Now, first is what anemia. I already mentioned this example, that is erythropoietin that can be produced. That with erythropoietin formation can be increased by using this technology that is related to anemia. Then asthma, interleukin, that is also a type of asthma where the problem is in taking of the breathing, and also the further not only the bronchitis, but the further next stage is also observed, where interleukin receptor is very helpful, that can be produced in using this technology. Then atherosclerosis, this is the word which is related to the blood vessels, where because of this is the condition where building up of the fats and cholesterol is there on the inner linings or the walls of the, and such type of substance are deposited on the wall of the blood vessels, or especially artery walls. And because of that, what happens, the arteries become narrow, and so it there is destruction in the blood flow, so blood cannot flow easily. Disturbance in the normal blood flow takes place because of this deposition of these fats and cholesterol, such type of substances on the arterial walls. And so in that case, this growth factor can be produced by using this technology. The next is what parturition, that is general, that is what childbirth. During that process, the relaxin is the protein which the word itself suggests, it relaxes the muscles of the pelvis and that particular tract which is used to give the childbirth. So it relaxes the muscles, so the delivery of the baby becomes easier with the help of, and that relaxin also can be produced by using this technology. Then blood clots, as I mentioned just now, that is tissue plasminogen activator. The example urokinase I already mentioned, that is very useful. So blood clots, because of the blood clots, what happens? It obstructs, or at the obstacle in the blood flow takes place, and that by producing TPA, that means tissue plasminogen activator, we can overcome this problem. And answer, I mentioned already about all these points, that is the interferon, tumor necrosis factor. Now just this point is what macrophage, macrophage means what, the role of macrophage is what, phagocytosis and pinocytosis. If you have word phagocytosis, means that if there is attack of any foreign body or the organism to the body, then what happens, these macrophages, they shows some type of inflammation, or sometimes by phagocytosis they engulf these bacteria or the foreign bodies, and there is destruction of the that particular um, uh, microorganism or any foreign body which is there in the attacked in the host. So these are playing very important role in the immune system. Then next is diabetes, already we mentioned insulin. Then emphysema. Emphysema is, some of you might be knowing also, that is chronic obstructive pulmonary disease. It is, pulmonary means what? It is related to the our lungs. Why? Lungs show the presence of, if you have learned the structure of lungs, it shows the presence of these air sacs. What we call these air sacs? The alveoli, right? So these air sacs, they become damaged, and these are stretched out, and generally that results into coughing, difficulty in breathing, and that all can is main cause, generally reason behind it is, common reason is observed as smoking, and it can be the genetic disorder also. So by producing antitrypsin, alpha antitrypsin, we using this technology, this problem can be overcome. Then I have already mentioned about hemophilia A and B, that these factors uh, are related to the genes which are responsible, the factor 8 for hemophilia B and factor 9 for hemophilia, factor 8 for A and 9 for B. And then hepatitis, already we have discussed about this, hepatitis B vaccine is produced using this technology. Now we will come to the next important role of Bacillus thuringiensis, which is related to biotechnology only. Now, what is this? In case of plant biotechnology, now we were talking so many examples about the animal biotechnology. Now, this is very special example that is involved in plant biotechnology, that is Bacillus thuringiensis. It's a soil bacterium and it produces a protein that has insecticidal properties. You know the meaning of insecticidal, the meaning of pesticide, insecticide, that means which kills the insects, which kills the pest, rodenticides which kills the rodents. So that is what sidal is, the killing effect. Okay, so which kills the insects. So this protein is produced by the soil bacterium. Okay, that is Bacillus thuringiensis, which has this insecticidal properties. And traditionally, the sprays are used to be in the form of sprays. But now, by using this technology, we have developed so many crops. The most common example, the first one, not first one actually, but the most commonly seen and known example of this is BT cotton. BT cotton means what? Biotechnology produced cotton doesn't mean that it is biotechnologically produced cotton, but that BT represents the name of this bacterium, this soil bacterium. B for Bacillus and T for Thuringiensis. So the first letter of each genetic as well as the genetic as well as species is used. So where you can remember it as the BT, that means for Bacillus thuringiensis. So this is the crop plants are now grown using some of, so again this problem is coming, just bear with me, I can't help. So I will try to come to that point. Okay, so this is what BT toxin. Now, what is, the meaning of toxin? Toxin is the word related to toxicity. That means harmful nature. Okay, which gives harms. So crop plants are now engineered to express BT toxin. Now crops are produced in such a way using this technology. By using this BT is what? This Bacillus thuringiensis bacterium. So Cry gene in BT produces inactive protoxins. Now try to understand what is cry. Okay, cry means crystal protein is produced using this technology, and that in BT, that is Bacillus thuringiensis, that produces inactive protoxins. Now try to understand inactive means which is not active in whatever the form it is right now present. And toxin means what? Toxicity. Pro is what? Previous. So previous form of toxicity means that is not at present in this situation. When it is a normal form, it is not toxic in nature. But when it becomes toxic, that we will see now. Then the mechanism of that BT we will see. What is the mechanism? BT produces the BT toxin, which are inactive protoxins. When an insect ingests it, inactive protoxin gets converted into active form due to alkaline pH of the insect's gut. And this leads to swelling of gut and ultimately death of insect. Now, what is done in this case, suppose this is a plant where this technology is used, where BT is introduced in the plant, and this is having this gene which produces the protoxin, that is which is inactive in the inactive form of protein, that protoxin. But when this insect feeds that particular plant crop, feeds on that crop, and it ingests that particular plant, then what happens in that? First of all, this in the plant it is in active form, and it is present in the plant, it is in the inactive form. But when it is ingested by the insect, then what happens when it is ingested by the insect? Because of the uh, alkaline nature of the gut of the insect, what happens then? Because of the alkaline nature of the insect, this inactive form of toxin then gets converted into active form, and because of that, this uh, swelling of the alkaline nature of this gut of the insect, it results into the swelling of the gut of the insect, and that results into the death of the insect. So this is what the mechanism of BT, that is the protoxin produced using this Bacillus thuringiensis, like cotton. These are some of the examples by which these crops are produced. That is BT corn, then like this BT. Um, if it happens like this again, I will just come out of the this thing, and then I will again. Okay. BT rice, that is the golden rice, very good example of BT biotechnology. You know, the color, it appears to be golden, and why it is produced, this rice contains vitamin A, and because of the deficiency of vitamin, which is present in rice as well as in some of the vegetables, like, which vegetables it gives? Carrot and all. So because of the deficiency, what happens? Blindness. And so to overcome this problem, so the rice with the high amount of vitamin A is produced by using this technology. Now BT cotton, what is the use? The long continuous threads, the food, what is the fruit, which is the product of this plant, that it shows the continuous long fibers, so that is economically very important. Then BT tomato, what is the use of this tomato? Its lifespan has been increased by introducing a gene, the enzyme which is responsible for the degradation of the cell wall, pectin of the plant, that fruit, that is reduced, its activity is reduced, so that it will remain fresh for a longer time. So the life shell of the fruit, that is the tomato, can be increased. Then brinjal, BT brinjal, then soybean, oil content has been increased. Then potato, the duration of storage is increased, and few more things. So these are some of the examples. In detail again we will go. The next part of this is gene therapy, which is the part of our DNA technology. Now gene therapy is nothing but the treatment of disease by replacing, altering, or supplementing a gene that is absent or abnormal, and whose absence or abnormality is responsible for the disease. So there are different definitions used for the gene therapy, but what is the main thing to be considered in this is what? Replacement of the defected gene, that you remember, so that that disease can be cured. Okay, so one more definition is there. Novel approach to treat, cure, or ultimately prevent a disease by changing the expression of person's gene. So this is one more definition. So that defective gene can be replaced by the normal one to overcome the problem. That you remember that. Now, gene therapy is the insertion of genes into an individual cells and tissues to treat a disease in which a defective mutant allele is replaced with a functional one. As I said, the defective gene might be because of the mutation or some other reason. That defective gene is replaced by the functional one. Then DNA is used as a therapeutic agent. Then genetic diseases, metallurgical disorders which are related to the blood, then immune related problems, then cancers. These are some of the examples where this technology, that is the gene therapy, is very important. So what are the types of gene therapies? There are two categories basically involved in this, that is the germline gene therapy and somatic gene therapy. Now the word itself suggests what is the somatic cell therapy and germline cell therapy. Somatic, which is related to the somatic cell, body cell, which can not be transferred to the next generation, and germline, that can be inherited. So these are the two types. Now few points which you need to remember about the somatic gene therapy and germline gene therapy you need to understand. Now, this somatic gene therapy affects only the targeted cells in the patient, and it is not passed to the future generation, because it is related to the only to the somatic cells, not the gametic cells. So if for that particular patient only it will be useful. If it is the inherited disease, then it is of no use. It the next in the next generation it cannot be passed, so it affects the targeted cells in the patient only. It is short lived because the cells of the most tissues ultimately die and are replaced by the new ones, because the after every particular duration each cell has its own life duration, so the new cells are produced continuously, so it's of short lived type. Then transporting the gene to the target cells or tissue is also problematic. Now, how it is problematic, when we will study in detail, then only you will be understanding. So we'll not discuss in on this part, which is not involved also in your chapter. So, but some disorders like cystic fibrosis or muscular dystrophy, that can be overcome by using this somatic gene therapy. Now, what is germline gene therapy? That results in permanent changes. Now, it's very important, results in the permanent changes, so that if it is inherited, means if it is the what you can say genetic disease, then that can be overcome, not temporary but permanently, so the next generation will not show this disease. So this is potential for offering a permanent therapeutic effect for all who inherit the target gene. Then possibility of in eliminating some diseases from a particular family, like suppose hemophilia, we were discussing just now, isn't it? This is a genetic disorder. So in such examples which are the inherited to the next generation, in that case this will be a very good therapy, that is germline therapy. So some of the people, there are different ways, controversies, so we'll not go to that controversies and all, but what is the main thing, that it can be the permanent solution, this germline gene therapy. Now, inhibition of gene expression. Now, how these genes are expressed to inhibit some of the cases. What are these cases? For example, mutated gene can be blocked at that DNA, RNA, protein level in case of mutations. Means the mutations which are giving negative effect, negative dominant effect, only in that cases it can be useful. For example, that can be useful in the cancers caused by inappropriate expression of a gene. If you come to know that this is the gene which is responsible for the cancer, then that can be inhibited, or this gene's expression can be blocked or stopped, or it can be you can see in this problem. Just a minute, we'll see. Okay, so the next example we will see, that is, these are some of the few more examples mentioned, that step by step I will explain these examples, and again in detail also in your some of the examples are coming in detail also in your textbook also. Now, this is gene therapy being used in many ways. For example, to replace missing or defective genes. As and when I require the, I think that explanation is needed, I will explain. But if you find that it is to be explained, you can unmute and tell me, or at the end we will discuss. Okay. To replace missing or defective genes. Then to deliver genes that spread the destruction of cancer cells. So, which speed up the activity of these cancerous cells. So these genes can be destructed by using gene therapy. Then to supply genes that cause cancer cells to revert back to normal, then that also can be done. The cells which are converting into cancer cells, then that can be revert back to normal cells. This also can be done. Then to deliver bacterial or viral genes as a form of vaccination. So vaccination also can be done by using delivering bacterial or viral genes. Then to deliver DNA to antigen expression and generation of immune response. Supply of gene for impairing viral replication. Viral replication in most of the cases, it spreads that particular disease. So that can be overcome by using this therapy. Then to promote or impede the growth of new cells. Now, this gene therapy, how to achieve the pharmaceutical effects? Some of the examples are given. Introduction of gene that makes cancer cells susceptible to anti-cancer drugs. When we use anti-cancer drugs, very easily these cancer cells will be susceptible to those drugs, and the further process of uh, formation of cancer cells will be stopped there itself. Then introduction of toxic genes whose expression may result into, okay, so toxic genes whose expression kills cancer cells. Then one more thing, cytokinins are very important in the immune system. So to enhance their potential can be done, which helps to kill diseased cells. So that also can be genes of the cytokinins can be expressed. Now these GMOs are mentioned where GMOs, that means genetically modified organisms. These are mentioned in your textbook also. Few more examples also I have mentioned, and these are in the textbook also. These are living organisms whose genetic material has been artificially manipulated in a laboratory through genetic engineering. This creates combination of plant, animal, bacteria, and virus genes that do not occur in nature or through traditional crossbreeding methods. So we use so many traditional methods, cross breeding methods, as well as hybridization and all these. These are very commonly used, traditionally used, but it doesn't involve this. These are produced in a different way. These genetically modified organisms, but they are not only useful produced, but how they increase the yield, and also they are resistant to drought. So drought tolerance capacity is more in such GMOs. Then they have enhanced nutrition. Nutrition is also more as compared to the normal one. Then in any other consumer benefit also. Now what are these benefits? Step by step we will see. Now transgenic plants. Traditional breeding programs involve sexual crosses, like hybridization and all. But now they give the high quality of present day plants. But this is one more important method where more recently biotechnological approaches have been applied to these plants to create genetic variations that are beneficial for mankind. Now what are these how these are beneficial? These are the examples of transgenic plants, no? Some of the examples we already discussed. So first plant was tobacco. More than 60 transgenic dicot plants and several monocot plants like maize, oat, rice, wheat are known. Tomato, soybean, potato, sugar, beet, grapes, brinjal. So these are some of the examples of transgenic plants. These are being looked up by bioreactors for molecular forming. Molecular forming means on the molecular level its production is increased. What is and for which use? In some cases they are used for the formation of drugs, in some cases vaccines, then antibodies, then amino acids. So and to as the for the immune system also, some of the drugs are produced by using this method. Now advantages we will see. First advantage in using this technology, that is food plants, is insect pest resistance. you know to reduce the use of chemical pesticides and to reduce the cost of food production, the use of Bacillus thuringiensis sprays is very, very effective, but limited because of low stability of the protein in the field. Previously you also might be knowing that sprays are used for the pests and insects, right? So these give harmful effects later to the plants. To avoid this, and also to reduce its cost, they are very costly also. This can be the advantage when if you use the insect pest resistance. We increase this capacity, we introduce the this particular pest resistance into the plant itself, then we can overcome this problem. Now we have already discussed this, that is the gene called cry gene present in Bacillus thuringiensis produces a protein that forms crystalline inclusions in bacterial spores, and when ingested by the susceptible insect, a combination of high pH and the enzyme proteinase of the insect's midgut processes them hydrolytically to release the core toxic fragments, resulting into the swelling and then death of the insect. We have already discussed about this in case of cotton, bt cotton. What happens generally? The bollworms, cotton bollworms, they are highly susceptible to these uh, bollworms, and when these bollworms feed the cotton ball, then this cryogen is already introduced using this technique. So when they eat this, automatically the swelling of the midgut takes place because of this protoxin that gets converted in active toxin, gets converted into toxic. Its toxicity is observed only in case of this alkaline nature, okay, of the gut of the insect. Now the next example benefit is biofortification, that means improved nutritional qualities. That means nutrition factor is considered here. So nutrient value can be increased using this technology. And just I mentioned the vitamin A, where in case of carrot and in some of the milk products, which is not uh, very easily available to the poor people. And so to solve this problem, what is now we have produced this transgenic rice and transgenic mustard. These are called as golden rice and golden mustard because of its color. Golden gold color is seen, so that's why, which are the examples with high amount of vitamin A in those plants. Then I mentioned the example of soybean, like that, oil palm, then rapeseed, sunflower, which are used for getting the oil. So the nutrient value, that means quality, is increased in these oil plants by using this technology by transfer of Arabidopsis genes. Okay, so these are the some of the examples to increase the nutritional value. Then in post harvest uh, thing, or this way also, the modification in the post harvest characteristics also very important. In case of tomato, I just mentioned, I was telling that the enzyme polygalacturonase is the enzyme which breaks down the cell wall constituent pectin. Now you know the cell wall of the tomato that contains the plant cell wall, which shows the presence of pectin, right, which shows the presence, and that pectin, what is the role of that cell wall, that enzyme? This is the polygalacturonase enzyme which breaks down the cell wall, and that leads to the softening of the fruit. That means the tomato becomes soft easily. But if we inhibit the activity of this enzyme, then what will happen? By antisense genes, these, its life shell, that means the cell wall will not degrade easily, and no, in the normally the shell life period is very less in case of the fruits like tomatoes and all. So in that cases, if we increase the life shell, that means the storage also can be storage period also can be increased, and there will not be loss economically also. The storage period also can be increased by inhibiting the activity of this enzyme. So the role of this enzyme is what? Degradation of the cell wall. But if we inactivate this enzyme, then automatically the uh, pectin, that means cell wall will remain fresh. So shell of the that particular fruit, that means tomatoes, will remain fresh for a longer duration. And so the name is flavor saver means flavor is also saved and they remain fresh for a longer time. So this is the best example. So post harvest, why it is called post harvest? After the production, what happens? Every plant crop is having its own storage period. So because of this pectin cell wall degrading enzyme, what happens? Within a short period they start degrading, the fruits get spoiled. So to avoid this, its life to increase its period of storage can be increased by using this method. That's why it is called as post harvest. Now the next is what? Plants as factories. Now they so many plants are used to get the more production of different products. For example, in case of pharmaceutical industries, biochemicals and vaccines are produced, and plants are potential, actually, these are considered to be the potential factories or bioreactors for getting high value biochemicals like starch, sugar, lipids, proteins, and few more chemicals are which are very important, perfumes, then adhesive compounds, etc. So these can be produced by you introducing the gene responsible for that, and the in more quantity these products can be produced. And currently, many novel products have been commercially exploited for their products. For example, super glue. You know the meaning of glue. What is glue? It helps to stick, or it works as a adhesive. So adhesive proteins can be produced by using tomato plant. So the genes are present in the plant itself. So that plant, that gene can be screened and that can be introduced. So it can be the natural glue. So biochemical glue is, is used generally in case of surgery. So this super glue, as I mentioned, super glue, it is mentioned because tobacco plant has the genes which encodes that particular gene responsible for this uh, special feature of, that is adhesive feature. So in that case, to uh, repair the surgery part, after the surgery that body part can be repaired easily by using this factor, isn't it? Then the next example, transgenic plants, that means containing oil. For example, uh, you know, cod liver oil is very very important, isn't it? Cod liver oil is having very medicinal importance and nutritional values and medicinal values also. In many medicines it is used. So once you know the gene responsible for that, that can be introduced into the plants, and the plants can produce the nutritional values as good as cod liver oil contains. Means the equivalent type of nutritional values can be introduced in the plants by using this technology. Then plants will produce the anti-malarial drugs like this by using this technology. Then opium poppy is used, you know, it's a natural painkiller, it is considered to be. So we can introduce the gene responsible for this particular value, that is the painkiller, and that can be introduced and used in different therapies. So these are some of the examples where, and just one more just I will mention and I will stop here, that is the plants producing edible vaccines. Already the vaccines, how they are produced, if you consider, if these plants only, if you introduce using this technology, then these potatoes, tomatoes, bananas, and all these are commonly used plants. They can be, these are very easily edible and consumed by them. So vaccines can be introduced in these plants. So transgenic plants can be produced. Now I will stop here and we will talk about the transgenic animals in the next session, so that we can have little discussion about it on this. So I'll stop the presentation, but if you have any doubts about the whatever we discussed, the different examples and all, we can discuss. 5 more minutes are there, we can